Welcome student, my name is Dr. Pidamri Thakur. I am working as an assistant professor in the University Institute of Biotechnology, Chandigarh University, Krua. So today we will discuss about microbes in human welfare. Now first of all we should understand what is microbiology. So the branch of biology which deals with the study of microorganism is known as microbiology, and the biosphere, that is the biotic and abiotic components, has a variety of microorganisms that exhibit beneficial activities, and they include small algae, fungi, bacteria, protozoan, mycoplasm and related organisms. A large number of microbes that help human civilization through their useful activities, and these activities are either of domestic, industrial or commercial importance. So it has several applied branches such as medical microbiology, food microbiology, industrial microbiology, etc. So we will understand how microbiology is working for these all industries. Now first we will see in the production of food. Now a number of microbes are involved in their production through the process of fermentation. So when we see food production, there also we see role of microorganisms. Now the use of microbes in food production is almost as old as human civilization. Traditionally, a number of microbes from the surrounding environment are being used in production of several fermented foods. But nowadays a few examples are we are going to discuss now. So some of them are for the production of idli, dokla, jalebi. These are very common food and everyone see these food everyday life. Common Indian delicacies which we use in our daily life, we can see there. And for this we will first of all study that how we prepare dough. So we prepare dough for dokla, and the dough for the dokla is prepared by mixing grain flour with buttermilk. Now the lactobacillus is from the microbiology, and lactobacillus is the microorganism. It brings about the fermentation process. Normally we do not understand what is happening behind that dough formation, but lactobacillus is that microorganism which is helping for the dough fermentation. Now microorganisms like bacillus species and candida species and saccharomyces species are involved in these processes of fermentation. So we should understand what are these microorganisms, what are their basic structure, what is their environment surrounding, what is their temperature, and everything we need to understand. So many lactobacilli are involved in the preparation of many common food like jalebi, naan, etc. and everyone is using it in their daily life, but they do not understand the microbiology behind these food preparation. Some microbes or their fruiting bodies are directly used as the source of foods. So that is also a rich source in protein. Now this term that is SCP, that is single cell protein, it is also utilized, and it is basically the dead and dried cells of microbes, and such as bacteria, alga, mold and yeast. These are also directly used as food in many cases in many countries. They are obtained by growing microbes in various groups of different substrates. So we can see there are many examples of these microorganisms. They are working for the production of food, for the production and preparation of fermented food in our daily life. These microbes include bacteria like Bacillus subtilis. So these are very common examples which you can see in your daily life. Fungi like species of Candida and Saccharomyces cerevisiae. Alga such as species of Chlorella. So they are very low calorie, sugar free, fat free and rich in proteins, vitamins, minerals and amino acids. So that is why we should prefer these food, and we should understand that why microbiology and why microbes are very important for all of us as a food product formation and as a food product. Now next we will discuss about the second very important application, and that is alcoholic beverages. Production of alcoholic beverages is very old phenomenon. Alcoholic beverages are produced by the fermentation of specific substrates, and they include liquor like wine, beer, whiskey, and everyone knows about these all liquor. So use of microbes in making fermented beverages is very old. It's since 700 BC. You can talk about Egyptian times, you can talk about Romans and Greek time. That time as well we were using all these technologies for the production of alcohol and beverages, but we do not know what is behind, what phenomenon is working behind the production of these beverages. But nowadays, when we are in modern era, we understand the role of microbiology for the production of these alcoholic beverages. So a number of strain, for example, number of strain of yeast, Saccharomyces cerevisiae, ellipsoides are used in industrial production of wine. So these are the identified and already utilized strain, and when you become a microbiology student, then you need to understand how these strains work for the production of microbiology, for the production of these beverages. Now different flavors of wine, they are also obtained by using different fruit juices, and these fruit juices and these flavors we can utilize by using microorganisms. Now beer is another alcoholic beverage, and it is very common alcoholic beverage all over the world. Now liquor obtained from fermented grain, mostly it is obtained from the barley, and suitable strain of Saccharomyces cerevisiae is used for this particular fermentation, for the fermentation of beer. Now it is produced through various steps, like first of all we will do malting, that is the first step of producing beer, then second we will do mashing, and then after that fermentation will take place. So this is how we produce all these beverages within laboratory condition. It is allowed to stand for a few days after this fermentation process, and then it is clarified, then it is carboxylated, and then it is filled in bottles, then it is packed, and then it is marketed. So this is how this whole industry work for the production of beer, and which is very common alcoholic beverage all among the common people. Now wine and beer, they are produced without distillation. Whiskey is obtained by fermenting mixed grains of corn, wheat, barley, and the product of fermentation is then distilled. So this is how we see the production of beverages when we see the food industry and the beverages industry. And as a microbiology student, you need to understand the basic phenomenon behind all these industries. Now very important application, that is organic acid fermentation, which is also part of our daily life. So we need to understand how it is working. A number of organic acids are obtained by fermentation using various microbes. So many microbes are there, you can see these microbes, these are organic microbes, organic acid microbes, and there are citric acid, Aspergillus niger, for example, gluconic acid that is produced by Aspergillus niger once again. Then we have fumaric acid that is produced by Rhizopus arisis, and these all microorganisms are helping us to give these organic compounds. So we need to understand how they will be produced. So for that purpose, it's important that you study microbiology in detail and you understand how these processes occur, because separate process has separate phenomenon within an industry. Then tubular tower fermenter, they are also helping for the production of these organic substances. Now next application of microbiology which we see in our daily life, that is vitamin production, and vitamins are very important part of our life. We know we need two types of vitamins, fat soluble and water soluble vitamins. Normally our systems are producing it, but if we are not able to produce these vitamins, and because it is part of our daily life, so we need to have some industrial product for the formation of vitamins. So these are required in very small quantities for normal growth and development of the body, but still they are required. So we need to produce these vitamins, and we need to think about vitamins, minerals for our daily requirement. So these vitamins can be water soluble and fat soluble. So when we see vitamin name like vitamin A, vitamin B, and vitamin C, vitamin D and E and K, so they may be water soluble and they may be fat soluble depending upon. So as you know, vitamin B and vitamin C, they are water soluble, and vitamin A, D, B, K, they are all fat soluble. So fat soluble vitamins we can get from the non-veg sources as well, but for water soluble vitamins we have to entirely depend on citric acid, and many other plant sources we have to depend on. So therefore it is the microorganism which can be the best source for the production of microorganism, because it is very important and beneficial part of human health. Therefore they are to be consumed through food or tablets. So if we do not consume them, then we can have some deficiencies, and these deficiencies can lead to the disease, and these diseases can be harmful for the human health. Now vitamins are manufactured by fermented technologies as well. So using different microbial sources we can produce vitamins. So name of vitamins produced by different microbial sources. So first vitamin which we are producing from the microbial source, that is vitamin B2, and its source we are taking Neurospora gossypi, that is the first source of vitamin B2. And then vitamin B12, that is also produced within a laboratory condition by utilizing Pseudomonas danificans. So that is the source. Then vitamin C, it is also produced by utilizing microorganism, and that is Aspergillus niger. So vitamins are produced by these microorganisms. So you can understand how helpful microorganisms are to us, because they are fulfilling our daily life requirements. Then we see the next application of microbiology, because we are discussing about microbiological applications, how they are helpful to the human welfare. So we will understand now by producing next they are really helping us. So that is antibiotic production. So there are some secondary metabolites which can be produced by utilizing microorganism, and the product of fermentation, you can say once again, because most of our products are produced by the process of fermentation, and they have therapeutic importance and they are used in medical treatment. For example, first example which I will take here, that is penicillin, and all of you might have heard about before. Penicillin and number of other antibiotics are used in control of infectious diseases. So it is recommended. Nowadays we say okay, antibiotics are very harmful, but still when we are suffering with very high yielding disease, so that time we have to take antibiotics because we do not have any other alternative. So in that case we have to consider antibiotics, but other chemical sources of producing antibiotics can be harmful. But if we have microbial sources of antibiotic production, so that can be useful for human welfare. So antibiotics are the substances produced in small amount by certain microbes, and it inhibit the growth of other microorganisms. So that is why we call it antibiotic, because they are inhibiting the growth of bacteria, and they can be antifungal as well. So if they stop the activity of fungus, then we can call them as antifungal. Now these antibacterial or antifungal, as they are in nature, and their nature is to stop the growth of other bacteria and to stop the growth of fungus, we can produce them inside lab by utilizing microorganism. So the first antibiotic which was discovered, because we are discussing about antibiotics, so you should know first of all who has discovered first antibiotic. So that was by Dr. Alexander Fleming in 1929. So he was the first scientist who discovered antibiotic, and he was working with the pathogenic bacterium. For example, he was working with Staphylococcus aureus, and while working with Staphylococcus aureus, since then a number of antibiotics have been produced and used therapeutically. So some common antibiotics and their microbial sources I will be discussing now. So one by one we will discuss these sources and their production. So first one is chloromycetin, that is the first and second antibiotic we will be discussing it. So it was produced from Streptomyces venezuela. Then erythromycin, that is also produced from Streptomyces, and then penicillin, that you know is produced from Penicillium, and streptomycin, that is produced from Streptomyces gracius. So many deadly diseases, for example plague, whooping cough and diphtheria and leprosy, and which used to kill millions of people all over the world during old times, now it is controlled by antibiotics, and we have successfully utilized microorganisms for the production of antibiotics, and which was a big discovery and a big great achievement in the history of microbiology. Now we will discuss about the next application, and that is for the production of gibberellins. As gibberellins, everyone knows it is a growth hormone, and they are a group of growth hormones, and they are mainly produced by higher plants and fungi to promote growth by stem elongation. So they are doing stem elongation, and first time it was isolated by two Japanese scientists, that is Yabuta and Sumiki in 1938, and they were produced and synthesized from the rice seedlings, and it was infected with fungus Gibberella. So we have got to know, we got this idea that if Gibberella can infect it, so that means Gibberella has some characteristics of producing gibberellin, which is our elongation or stem elongation hormone. Now about 15 types of gibberellins have been isolated from Gibberella fusiculae. So gibberellins have many practical applications and we are able to produce it from the microorganism. Now they are used to induce parthenocarpy in apple and pear, etc. They are used in breaking dormancy and inducing flowering also. So we can artificially induce all these things among flowers and we can utilize it. So this hormone we can produce from microbial source. Now we will see the next application. So if I will talk about next application, it will be enzyme production. So enzyme production is very important application because enzymes are basically biocatalyst and they have property of accelerating a rate of reaction, and they accelerate all the biochemical processes in living organism. They speed up the reaction by lowering activation energy. So a number of hydrolytic enzymes that degrade starch, protein, fats and pectin into simple compounds are known traditionally, and for this we are using amylase, papain and pectinase, and they are utilized for food processing. And in recent years many more enzymes are being produced, and for getting cheese, butter, sweetness of confectionaries, animal feed, then soybean milk, then modification of food, gums, etc. Now we will discuss more example about the enzymes which are produced from the microbial sources. So first enzyme which is produced by the microbial sources is invertase, and it is isolated from Saccharomyces cerevisiae. So now we are nowadays utilizing Saccharomyces cerevisiae for the production of invertase enzyme, and now we are utilizing Sclerotiana libretine for the production of pectinase. And because we have to see industry production, so inside industry microorganisms will be the best source for the production of enzymes, and that is why our more focus is for the utilization of microorganisms for making more and more industrial products. And one more enzyme which we are producing, that is lipase, and it is very important enzyme. It is produced from Rhizopus species. And next enzyme which we are producing nowadays is cellulase, and we are producing it from Trichoderma. And if we are talking about next industry, that's all about enzyme industry. Now we will talk about dairy industry. So various products obtained from milk in dairy industry using microbial species we have to discuss now. Some of these products are very common to everyone and everyone knows about it, that is cheese, yogurt, buttermilk, paneer, etc. Now species of Streptomyces, Penicillium and Lactobacillus are commonly utilized for the production of these products. Now, all the domestic level preparation of fermented milk products started in early period of human civilization. So everyone knows about it because we know how to utilize all these things at home, but we do not know what is basic microbiology behind that and which microorganisms are working for the production of curd, for the production of milk and everything. Now curd and buttermilk were produced using lactic acid bacteria. So this everyone knows, and maybe might be some people do not know, but when you study microbiology then you have to read about it. And the microbes can be helpful for the production of these all household things like milk, butter, and then we are producing at home curd, then we are producing at home many other things we are producing, and all these are the products of microbiology. So as we know that microbiology are really beneficial for the human being, for the welfare of common people, so we need to understand what is basic microbiology. Now in continuation with the application of microbiology, now I will tell you about the next application where we are utilizing microbes. So next application of microbes will be in sewage treatment. So sewage treatment is such a big problem, so we have to solve that by utilizing microbiology. Now we know that large quantity of waste water, it is generated every day, and it is generated in all the cities and towns and even nowadays in villages as well. So this waste water is also called sewage. Now sewage water usually contains high level of organic matter, human excreta and domestic waste and microbes. So microbes can be pathogenic, and it is essential to remove organic matter from the waste. And if we know that how to utilize microorganism, then we can utilize these microorganism for improving that wastewater and to create and to convert that wastewater to some useful water. So that will be the biggest help for the benefit of mankind and for the usefulness of human being. So I think that's all about the applications, and we have studied now so many applications about microorganisms, where and how we can utilize microorganism for the welfare of human being, mankind. So many examples as well we have discussed here. So rest of the things we will discuss next time. Thank you so much everyone. Thank you.